Wonderful morning or evening to all of you. I am Pradeep. I am presenting our work titled Fuzzy Entropy as a measure of EEG complexity during Rajayoga practice in long-term meditators. My co-authors are Professor A.G. Ramakrishnan and Dr. Kanishka Sharma. This work was carried out at my laboratory, Department of Electrical Engineering, Indian Institute of Science, India. In spite of extensive study on meditation EEG, not much is known about the process and impact of meditation on the brain. EEG recorded during the meditation may provide information more than the normal consciousness or awareness. There are a number of publications on meditation based on spectral analysis of EEG. It is suggested that the elevated states of consciousness like metacognitive functions and self-awareness are associated with higher entropy. Entropy measures have been used in the studies related to epilepsy, Alzheimer's disease, and anesthesia. Studies have been conducted on Rajayoga practice of Prajapita Brahma Kumaris World Spiritual University, and I shall go through some of the reported results. It was observed that the networks were activated across delta and low alpha frequency during seed stage meditation under Rajayoga practice. Study on long-term Rajayoga meditators reported higher frontal and parietal cortical asymmetry and increased band power in theta and alpha band. Neuroimaging study on long-term meditators of Rajayoga practice reported an increase in white matter integrity in corpus callosum segments. In another study on the same dataset used in this work, it was observed that the mean and variance of the distance between covariance matrices of successive epochs decreased during meditative states as compared to non-meditative states, indicating uniform interrelationship between the channels in the frontal lobe. Data was collected at Brahma Kumaris headquarters at Mount Abu as given in figure 1. Baseline recording was carried out for approximately 3 minutes, while the subjects were asked to relax with their eyes open before meditation. Baseline was followed by the peaceful soul consciousness state of meditation, wherein the subjects meditated by focusing their attention on the positive thought about themselves as peaceful souls. This lasted for around 10 minutes. This was followed by another intermediate baseline for 3 minutes. After intermediate baseline, subjects were asked to delve themselves into angelic state of meditation for 10 minutes, where they are supposed to attain a blissful state. A final baseline was recorded for 3 minutes. Data comprised of 15 healthy right-handed subjects who practiced Rajayoga meditation regularly for a period of more than 10 years. Rajayoga practice is done with eyes open, which makes it unique among other meditation techniques. The fundamental difference between these meditative states is believed to due to self-observing experience in peace meditation and the state of being detached from the physical body, relationships and material assets during the angelic meditation. The data was recorded with ANT neurosystem with 64 channel waveguard cap according to 10-10 system. Sampling rate of 1024 samples per second was employed during the recording. Post-recording, line noise of 50 Hz was filtered using notch filter, followed by 0.5 Hz to 45 Hz bandpass filtering, and independent component analysis was applied on the data using EEG lab. Grouping the channels was carried out as given in figure 2 into 5 broad regions, namely frontal, frontocentral, perietocentral, occipital, and combining temporal right and left channels as temporal region. Abstract of the proposed methodology is given in figure 3, which shall be dealt with in detail later. Non-overlapping epoch of 0.5 second each consisting of all the channels of a lobe is considered to obtain the feature for each epoch. Entropy of EEG quantifies the amount of randomness in the signal pattern, which is equivalent to the amount of information. The proposed work is based on the minimum variance modified fuzzy entropy feature, which is used to distinguish between the different meditative and non-meditative states. As mentioned in the previous slide, xk is the kth epoch data matrix of any given lobe and state, that is baseline, peace meditation, intermediate baseline, angelic meditation, and final baseline. M is the number of channels in the data matrix which varies with the lobe. N is the number of samples in each epoch and is the same across the states and lobes. And K is the epoch number. The relative energy function given in equation 1 maps the finite set of samples of data matrix between 0 and 1. The relative energy given in equation 1 is used as a new membership function to calculate the entropy as in equation 2, and hence the name modified fuzzy entropy. The modified fuzzy entropy is denoted by HIK corresponding to ith channel, kth epoch of a given lobe and state. Each epoch data matrix results in a vector of length equal to number of channels. By concatenating all the vectors hk of k epochs, matrix H is formed for each of the segments, namely baseline, peace meditation, and so on for each lobe. A scaling operation is performed on matrix H to increase the distinction between the meditative and non-meditative states. First, the modified fuzzy entropy is divided by the minimum value across all the epochs of the same channel. Second, the power of a new modified fuzzy entropy values are divided by the variance of the new modified fuzzy entropy values of the respective channel. Hence the name minimum variance modified fuzzy entropy feature. The time course feature values are averaged across all the channels to obtain a row vector for which the mean is calculated for a given subject. Wilcoxon test was used to check the statistical significance of the results obtained. Test revealed that the mean of the feature extracted was significant in the frontal region of the brain. Figure 4 is the plot corresponding to the mean with standard error mean of the meditative and non-meditative states across all the 14 subjects, each figure corresponding to lobe of the brain. The pink dots represent the mean and the bars are the standard error mean. Table 1 and 2 are the z values and p values of the frontal lobe compared with all the meditating and non-meditating states. Figure 5 is the univariate scatter plot of the mean of all the meditative and non-meditative states in the frontal lobe EEG. Each point corresponds to a subject. Black line represents the mean and the grey box the standard error mean. Studies conducted by Vivo and group and Venugopal and his group reported increase in entropy during Vipassana meditation. The frontal lobe is involved in the execution of behavioral responses and complex mental responses to environmental challenges. It is also considered to represent the internal states and accumulate external information. The higher statistical significance in frontal lobe compared to other lobes need to be investigated further. Comparing the results reported in the present work with the available literature is difficult, given that there are very few antecedents of scientific papers investigating the fuzzy entropy measures for meditation data. A primary source of confusion is the difference in the traditions of meditation types that are investigated. Also, the neural activity during meditation is confounded by many factors and hence the results may not be consistent with other meditation methods. To conclude, the mean of the entropy feature extracted is higher during the meditative states as compared with non-meditative states in the frontal lobe, indicating a non-pharmacologically induced brain state of high entropy. Our future work includes analysis of the same features for different frequency bands of interest, namely delta, theta, alpha, beta, and lower gamma bands. We thank the support extended by Spark wing of Brahma Kumaris organization by lending their EEG system for the study and the subject who participated in this study. I also thank my co-authors for their contribution towards this study. References slides will list our references used. Thank you for listening. I shall take the questions, if any.